Many cells can precisely control the time of their own death through apoptosis, also known as programmed cell death. Apoptosis has several important functions in our body. It is involved in embryonic development, in which selective cell death shaped the adult body. Apoptosis can also be triggered by irreparable DNA damage. During antibody development in the thymus gland, the plasma cells that generate anti-self antibodies are eliminated by negative selection through apoptosis. Apoptosis is also involved in the shedding of cells of the uterine wall during menstruation. The role of apoptosis in embryonic development was first studied in the soil worm C. elegans, which undergo exactly 131 times a programmed cell death at precise cell lineages. Apoptosis in C. elegans involves three key proteins, Z9, Z4, and Z3. The pro-survival protein Z9, located in the outer mitochondrial membrane, serves as the master regulator of apoptosis. Under normal conditions, Z9 inhibits the pro-apoptotic protein Z4, thereby keeping the cell alive. When a death signal such as EGL1 is received by the cell, Z9 is inactivated, relieving its inhibition of Z4. Z4 is an adapter protein that activates Z3. Active Z3, which is a protease, triggers a cascade of reactions leading to activation of nucleases and other proteases, ultimately triggering apoptosis. Apoptosis in mammals is largely carried out by a type of protease known as caspase, which stands for cysteine aspartic proteases. Cysteine is located at the active site, and the enzyme cleaves C-terminus aspartate residue. There are two types of apoptosis pathway in mammals: the intrinsic pathway, which begins within a cell, and the extrinsic pathway, which begins outside a cell. The intrinsic apoptosis pathway may be triggered by DNA damage, metabolic stress, developmental signal, or accumulation of reactive oxygen species, abbreviated as ROS. Similar to the mechanism of C. elegans apoptosis, mammalian apoptosis is also regulated by a pro-survival protein BCL2, which normally inhibits the pro-apoptotic proteins BAX and BAC. Under stressful conditions, BH3-only proteins such as BAD and BID inactivates the regulator BCL2, thereby relieving its inhibition of BAX and BAC. In addition, reactive oxygen species such as hydrogen peroxide can cause the phospholipid cardiolipin located on the inner mitochondrial membrane to undergo peroxidation, also activating the pro-apoptotic proteins BAX and BAC. Active BAX and BAC changes the permeability of the outer mitochondrial membrane, allowing cytochrome C found in the intermembrane space to diffuse out of mitochondria through permeability transition pore complex, abbreviated as PTPC. When released into the cytosol, cytochrome C interacts with the monomers of the protein APAF1, which stands for apoptosis protease activating factor 1. The binding of ATP triggers the formation of a multi-complex protein apoptosome, composed of 7 APAF1 and 7 cytochrome C molecules. The apoptosome provides the platform on which the protease pro-caspase 9 can dimerize to generate active caspase 9 dimers. Caspase 9 catalyzes proteolytic activation of caspase 3 and caspase 7, ultimately leading to cell death. Notice that APAF1 serves as an adapter protein that binds to cytochrome C and activates the pro-apoptotic protease caspase 9. On the other hand, the extrinsic pathway is triggered by extracellular death signals, which binds to cell surface receptors. The death signal Fas ligand binds to Fas receptor on the plasma membrane, which triggers the death domain to interact with cytosolic protein FAD, which stands for Fas-associated death domain, which in turn activates the cytosolic protease caspase 8. Similarly, the death signal TNF-alpha, which stands for tumor necrosis factor alpha, binds to TNF receptor, triggering interaction of death domain with TRAD, which stands for TNF receptor-associated death domain. TRAD also activates the cytosolic protease caspase 8. Active caspase 8 can trigger the release of cytochrome C from mitochondria, resulting in apoptosis through caspase 9. Caspase 8 can also directly activate caspase 3 and caspase 7 to trigger apoptosis. Phagoptosis is another type of cell death caused by the cell being phagocytosed or eaten by another cell. Phagoptosis is normally caused by an eat me signals that a cell exposes on its surface. In one type of mechanism, the activation of scramblase by calcium cations triggers the externalization of phospholipid phosphatidylserine on the plasma membrane. In this case, phosphatidylserine serves as an eat me signal, which triggers phagocytosis of the cell.